Hello, my name is Abigail and I chose to research diagnostic testing for rare idiopathic diseases. So diagnostic testing is accomplished via genome sequencing, which is a type of precision medicine, and idiopathic diseases are those that do not have any genes linked to the disease. Because of this, they are very difficult to treat because the only thing that you can assess are the symptoms. You don't know what's causing them, and because you don't know what's causing them, you really have no way to accurately treat them. And this is where genome sequencing comes in because it develops those novel relationships that were previously unknown between genes and the genes that cause this disease. Examples of some rare diseases are Rotor syndrome, Wiskott-Aldrich syndrome, Roberts syndrome, and cat eye syndrome. So genome sequencing, when performed correctly, has about a 25 to 30% success rate. And this seems like it's relatively low, and it is. The reason being is that just because a relationship between a gene and a disease has been identified, that doesn't mean that there's a viable way to treat it, which is why those numbers are fairly low. But when a relationship is identified between a gene and disease, and there is a method of treatment available, obviously the prognosis is very hopeful. The IDIOM, the Idiopathic Diseases of Man, their goal is to discover novel relationships between genes and diseases, and this is accomplished via molecular genetic diagnosis. They come up with treatment plans, and this is integrated with clinical assessment and multidisciplinary case review to make sure that they're looking at this disease and the genes associated holistically and gathering as much information as possible. They've done multiple studies with high success rates, and there have been, via some of the studies they've done, they have developed new relationships that were previously unknown between genes and diseases. There are criteria that the IDIOM uses to establish for candidates, and I'm going to read those off to you. They're quite in-depth. Number 1, the patient has a grave or serious condition that is undiagnosed despite extensive medical and genetic testing and evaluation. Number 2, the patient's condition is potentially actionable or amenable to treatment, and again, this is subjective based on the physician review panel that typically excludes individuals that have severe dysmorphologies. Number 3, the condition seems to be genetic in origin. Number 4, the patient's anticipated life expectancy is consistent with the study timeline for sequencing. And number 5, the patient has a physician champion who is willing to work with the research team and take responsibility for returning the genetic results to the patient. So now let's get into the process. How does genome sequencing happen? Once those 5 criterion are met, the next process is gaining consent from the patient and the patient's family. They're going to look at biological family only, and they're going to draw blood from the proband, which is the patient, and the patient's family members. This blood is going to be assessed for a few different things. Number 1, whole exome sequencing is performed, and this is to detect coding variants. Then you are going to look at whole genome sequencing, and this is used to detect copy number variants. Now, in genetics, obviously everything that you're looking at is very minute, but there are even more minute things that you're going to look for in genome sequencing. And this would be if there is one single insertion or one single deletion. These are obviously very hard to pick up using the whole genome or exome sequencing, and this is where Sanger sequencing comes in. It is algorithms developed specifically for picking up on those very minute changes in that DNA code and sequence. After the genome has been reproduced in the lab via the blood work, the information is presented for dissemination before a clinician scientist review panel that includes the patient's physician champion. They're going to address any potential issues, and they're also going to address whether or not the findings call for alteration of the patient's treatment plan. Typically, it will call for alteration of the treatment plan because it's an idiopathic disease that we're dealing with. So we were kind of just stabbing in the dark before we did genome sequencing. Ultimately, the research team can present the information. Here's what we found, here's the relationship between this disease and these genes. Ultimately, it comes down to what the physician wants to do, the patient's physician champion. He or she is the one who is going to decide to move forward with care or alter care in any way. Typically, the findings that are discovered are centered around neurobiological disorders, but it also includes hematological or oncological findings, dealings with allergy and immunology, cardiology, or gastroenterology. Those are the main categories of disorders that are discovered via genome sequencing. And genome sequencing is very helpful when you can come to a conclusion about what's causing this disease, because then you can move forward with effective treatment plans. More specifically, obviously genome sequencing is new, so there are avenues to develop this research further. More specifically, something that I looked at was juvenile idiopathic arthritis. More specifically, systematic juvenile idiopathic arthritis, abbreviated SJIA. The study looked at 770 children from 9 different nations. Using those children, obviously they were all eligible and willing and able to participate in this study. They had a certain threshold, and they set this threshold based off of findings from the International Childhood Arthritis Genomics Consortium, and they examined their major histocompatibility complexes and the loci on chromosome number 1, and each of these findings showed that SJIA, systematic juvenile idiopathic arthritis, exceeded the threshold for genome-wide significance, meaning here's the threshold, here's the significance of SJIA, meaning there is ample room for future research. Overall, genome sequencing still has a lot of kinks to work out because it is so new, and again, there's only a 25 to 30% success rate, but moving forward, I expect to see much more development in this field.